give everybody an overview of the personalized precision nutrition, which is an emerging area of research, which has a lot of, what shall I say, potential to help us manage diseases as well as prevent diseases. So I'll just go through the background, what we know as of now, then what is emerging in this area and how we can move forward, as well as the opportunities and challenges, since it is an emerging area. So we all know that the food and nutritional sciences have provided a lot of scientific evidence in the area of the role of nutrients and non-nutrients. We know that they promote and maintain health and well-being, they help reduce the risk of diseases, and they facilitate the management of diseases, both among individuals and communities. This is a well-known fact for all of us in this area. We also know that diet is a key determinant of health, and the link between food availability and consumption styles to health and life expectancy and specific disease patterns over the last 3 to 4 decades has been well documented with extensive research evidence from diverse sources across the world. You have cross-sectional studies, you have longitudinal studies, various types of studies, intervention studies, and this has helped us to understand the link between the two, how diet is really fundamental to health. But much of the mechanism of action is not fully known. We are still trying to understand how exactly this operates. And many of us would be aware of the fact how the approach to diet is very different from the approach to drugs, because drugs are single molecules. Of course, they do interact both among themselves and with the food. But single compounds are easier to analyze and what shall I say, understand what we call the exposure. The effects of exposure are easier to understand. The effects of treatment are easier to understand because we can assign the results to one specific molecule, but that's not the case in diet. Now, we also know that traditional dietary practices are common both in India and other countries and tribal communities across the world, whether it is America, Africa, Asia. They've all been practicing various types of either herbal medicine or using food as a therapy. So we need to recognize that these traditional practices are used in many places, including our country. And here also, the diet and its components are considered fundamental to prevention, therapy, and management of diseases. So we find that this approach has been there, but the science behind it we've not understood. There's some empirical evidence because you have practitioners of Siddha, Ayurveda, etc., who use food sources, both plant and animal origin, as well as microbes, because fermentation is an important technology which they use to produce the medicines. What exactly determines health? Two major factors determine our health. One is our genetic makeup and the other is our environment in which we live and we operate. So when we talk about environment, we have the physical, which is the air, water, soil, and climate, all of which are affected by pollution. Then we have the biological factors where we have diet, microbes, as well as our physical activity. Then you have the emotional or psychological factors. We have the socio-cultural context in which we operate, and economics, trade, and markets, which determine how much of food is accessible, what type of food is accessible to us is determined by those. So here in this talk, we are going to focus more on biological, particularly the diet and microbes. I will not be touching upon physical activity at all because that's, a lot of work is going on in that also, including sleep. There's a lot of studies on that. So here we will focus on the diet aspect and a little bit about the microbes. Now, diets are no longer evaluated only by their macronutrient and micronutrient content. Of course, they are very important and they are fundamental to our health, but we know that additional components play a role. So nutrients play specific metabolic functions, which helps to maintain our cells, tissues, and systems as a whole. The non-nutritive bioactive components, which have become the focus of attention in the recent past, over the last two decades, I should say, or 2.5 decades, they facilitate physiological functions, they promote health, and they prevent disease. And this is what had been practiced in the traditional medicine. So, but the scientific evidence for that has become more apparent recently. And bioactives in foods can be derived from micronutrients, from phytochemicals, which of course originate from plants, zoochemicals, which originate from animals, botanicals, which come from herbs, and some of these herbs may be non-food herbs. Then we have the prebiotics, and then we have the microbes, which are the probiotics. So all of these can strongly influence our health. So what is emerging today? What exactly are we seeing today in this area of nutrition research? We are seeing in the larger context, we are seeing the health impact of diets. Over the last 20, 25 years, there's been a rapid dietary shift. And in our country, over the last 10 years, I should say, increasing popularity of fats, meats, soft drinks, sugary and salty foods, which we think are the culprits for the various problems we face today. Simultaneously, there has been a decline in the indigenous fresh foods, which has been part of our traditional diet, which include the healthy snacks, the beverages, and fresh fruits. The other thing is there is a decrease in the diversity of these foods itself. Even when we think of indigenous fresh food, the diversity is coming down. We are not getting the although India is blessed with such a good wide variety of agroclimatic zones, and it's one of the richest biodiverse sections in our Earth planet. In spite of that, biodiversity is going down. Even if we take the cereals or pulses, the number of varieties are decreasing. Now there is a revival. Added to that, we have the problem of urbanization, globalization, supermarketization. We know that once you go to the supermarket, you end up buying foods which you really don't need. So this is a problem. Then recently we have this media-based food culture. So a lot of hype is created, and then people can get misled by that without having an informed choice, a scientific basis for their choice. And this media-based food culture is, in my view, can be for the good, but sometimes it can also create a situation where some foods which are not really balanced may be promoted. There is need for caution in this area. If we look at the health projections, it's really very depressing because we are projected to have the largest burden of diabetes mellitus and coronary heart disease in another 3 years. Imagine, we are in 21, almost end of 21, we are into 22. We will be exceeding China, as you can see in that bar chart. But along with that, we have very good things. We have increasing life expectancy. Today, Indians are living longer than they used to, say, 50 years ago. And therefore, there's an increase in the aged population. So this means there is an added burden of chronic diseases, because as we age, our susceptibility to chronic disease advances. And so both of these together are very significant, and we need to make sure that people who live longer should have quality life. There's no point in living longer if they cannot have quality life, they cannot maintain their health as much as possible, or as much as their genetic potential allows them. WHO statistics is also fairly alarming, and it's been shown that since 1975, obesity has tripled, diabetes has quadrupled, and high blood pressure is doubled. So we have a situation where 1 in 11 adults in India have diabetes. 1 in 5 people with diabetes are greater than more than 65 years old, and 1 in 2 Indian adults with diabetes are undiagnosed. And the story with respect to hypertension is fairly similar. We have more people affected in the urban areas compared to rural areas, and older adults about 25 years do suffer from hypertension, and we are close to the global average, although we are less than that. We are heading there. Now, why this situation is there? Simply because it's not a single factor which is affecting the etiology of the disease. It's a multifactorial nature of these conditions. And often there is difficulty in pinpointing the exact etiology for these diseases. And recommendations for healthy eating guidelines is often obtained from epidemiological or large clinical studies, which sort of doesn't give you the granular picture, the details as to how it can be effective in some people and not so effective in others, and not at all effective in certain others. So averages or generic cutoff points are made in an attempt to give nutritional advice at a population level. So these are only guidelines, but if we want more precise response to diet or to exercise, then we need to have more personalized approach, a more detailed approach, which tries to understand holistically the conditions which that person is actually suffering from, what is his genetic potential and propensity for a particular disease, and how we can manage that. This, as mentioned earlier, this is a practical generalization which we've made from the epidemiological studies. But in this practical approach, we have shortcomings because it fails to capture the individualized nature of the biological effects of nutrition. None of us respond the same way to the same plate of idli and sambar. We all have different responses. although the composition is exactly the same, each of us will respond to it differently, and the same person will respond to it differently on a different day. So this is the problem. And this does not take into account the inter-individual variability, how we each of us vary and to what extent we vary. And variability is known to exist in response to the same dietary intervention with respect to body weight, postprandial glycemia, and physiological response to salt. It's very well known. Salt restriction helps some people, it doesn't help everybody. In some cases, people who are on medication, they need to add salt to their diet, especially among the elderly. Caffeine metabolism, vitamin metabolism, and many others. So we don't know. We are still looking at that, and we need to understand how the responses differ. And the factors which are responsible for this variability include gender, ethnic origin, our genetic makeup, metabolic traits resulting from the interaction of the genes with the food, the environment, the microbial composition, the microbiome, and probably yet undiscovered factors. We don't know. Probably another 5 years, 10 years, we will understand these factors more. So the concept of precision nutrition has been proposed, and we are into this area today. What is this precision nutrition? It is an approach where the individual or stratified level is put forward as an answer to this problem, because we find unresponsiveness in many individuals. The diet guideline which we provide doesn't work for everybody. So generally we tend to blame the patient or the individual or the subject. So now we need to relook at that. This is a new discipline, relatively new discipline. There is no universally agreed upon definition, as Professor Honorable Vice Chancellor Dr. Premavati mentioned, okay? And it's founded upon the concept of biological variability between individuals in response to nutrition. We all know one man's food is another man's poison. That is one extreme saying. But we also know that we are highly individualistic. You can, in a single family, you can take. We have different responses to the food. Some people get diabetes, some people don't get diabetes. Even within the family, there is high variability with respect to weight gain, hypertension, etc. So the hypothesis is that if the variables which are responsible for causing this variation and their effect on a desired outcome variable is known to us, then this outcome variable can be predicted, and this can be translated into nutrition advice for that person or for that subgroup. And therefore, we are likely to see a better response. And therefore, that patient or that small group of people will benefit from that advice in terms of a favorable outcome, favorable response, or a better management of the disease or prevention of the disease. So precision or personalized nutrition is an emerging therapeutic approach which takes into account the individual's genetic and epigenetic information, as well as their age, gender, or particular physiological and pathological status. It has tailored nutritional recommendations, and this is based on the differential response to the certain individualized food-derived nutrients, which arise due to the interaction between nutrients and biological processes. These include the interactions between internal factors, which include genetics, the microbiome, metabolome, as well as external factors like dietary habits, physical activity, and even sleep. When we look at the basic elements for precision nutrition advice, essentially, I put them into 3 columns deliberately. One is relating to the diet. Dietary survey, all of us know how it is done. So the dietary intake, we have different tools to do that. The meal content, the meal timing is important. Then we have the personal details, which include, all the anthropometry, physical activity, other details also we collect about the medicines they take and the history, etc. Then on the other side, you have all things relating to the clinical and biochemical. This is what we are doing. We are looking at the clinical parameters and the biochemical parameters. You have, we measure the glycemic response, we measure the lipid status, we look at the liver function tests, so many we do. But in precision nutrition, the four important things which need to be looked into, one is the genetic makeup, the nutrigenomics, the metabolomics, and the microbiome. All of these play a significant role. We will see how. So precision nutrition has developed because of the phenomenal growth of other sciences, namely molecular biology and genetics. As you know, the biotechnology department, when I was a student, there was no biotechnology department. There was only botany and zoology. Then after that, biotechnology came, molecular biology came, and the genetics was always there. But the detailed look at genetics, that is the DNA sequence, RNA, all that came much later. A better understanding of the metabolic functions of nutrients and bioactives in cells and tissues, and the central role of microbes in our systems. This microbe, particularly, we are now understanding in greater detail, and it's one of the thrust areas of research today. Lots of laboratories all over the world are looking at this. And all of you will also know that although the research is still going on, but the industry is hopped onto it, and you have a whole lot of products in the market which are supposed to sort of balance the microbes in our systems. So this is another problem which we face today. Because of the growth in these sciences, there's an increasing understanding of the genetic variations across the human genome. How? Again, Professor Premavati had mentioned all these, the various techniques used were genomic sequence analysis. So what do we find from this, from the genomic sequences? The role of the genetic variants. So these fall into three types. You have the single nucleotide polymorphisms or SNPs. You have the copy number variations, which are called the CNVs, and other structural variants. So if we study more of biotechnology, we'll understand what this means, because small variations occur in the sequence, and these are found to be clustered in various areas, and it's possible to identify such variants. We have the epigenetic characteristics, and then we have the gene expression patterns in response to the diet, in response to the environment, etc. So we will look at some more details because we need to understand these terms. Some definitions. What is epigenetics? It is the inheritable and reversible processes which regulate gene expression without concomitant changes in the DNA coding sequence. So the DNA does not change. The DNA remains the same, but certain aspects relating to gene expression change. You have nutrigenetics, which is the science which studies the effect of genetic variation on the dietary response. Nutrigenomics is the science which studies the role of nutrients and bioactive components in gene expression, and therefore on the proteome and metabolome. Because from the gene, you get the mRNA, and from the mRNA, you get the protein. And then from the protein, you get the metabolites. So that is the sequence. Proteomics is the large-scale study of proteomes, which is the analysis of entire protein complement of a cell, tissue, or organ under a specific defined set of conditions. So if you're looking at one particular cell, you want to know, and under certain conditions, what are the proteins which are getting expressed. As you know, millions of proteins will be expressed. So this is how it is. So we want to look at, instead of looking at a single protein, we are looking at multiple proteins. Metabolomics is the analytical profiling technique for measuring and comparing large numbers of metabolites which are present in biological samples. So instead of looking just for example, the cholesterol, for example, when we say the LDL cholesterol, we'll also look at either the protein or certain other components, and VLDL, we may look at the fractions or details of those, the protein present in those. So metabolomics deals with that. Omics research has today become, again, very important because it has implications for nutrigenomics, proteomics, and metabolomics, all of which throw light on the specific interactions of nutrients with genes, proteins, and metabolites. So if nutrients are affecting the genes, it is going to affect the mRNA which is being synthesized, and therefore the proteins, and therefore the metabolites. It also provides information on individualized nutrient requirements, the digestion process, the absorption process, and metabolism process. Simple example would be, for example, any of the binding proteins which are involved in absorption, like for calcium or vitamin E. If there is some change in that, obviously that particular vitamin or mineral is not going to be bioavailable for that person. That's just a simple example. And now the gut microbiome, this is another big area. Each person's body is home to diverse microbes. This is something all of us know. Humans have about 10 to the power of 12 parenchymal cells, but we have much more microbes in our skin, in our mouth, in our gut. And the gut has, I think, 800 species is a sort of underestimate. It's likely to be more, with predominance of the obligate anaerobes, which includes the bacteroids, the Clostridium, Lactobacillus, E. coli, Bifidobacteria. And today we understand it's not a single group which affects, it is how they all live together. Just like in our community, how we can live peacefully or we can create problem between ourselves. Same way, how these are balanced out, how they interact with each other, and therefore they help the host. That is the area of intense study today. And it's also known that dietary factors strongly influence the composition of the gut microflora. So this is an intense area of research. which components in the diet affect, how do they affect, what is the consequence of that, how does the favorable microflora influence the functioning of various physiological systems. These are areas of study. Today, the probiotics was defined several years ago, 1995, but now the gut microbiome, very recently there was a paper, I've given it there in the list of references in the abstract. It's a larger concept. So today, the gut microbiome is considered a characteristic microbial community for each person, which occupies a reasonably well-defined habitat, has distinct physicochemical properties, encompasses their entire theater of activity, which results in the formation of specific ecological niches. This is what I said. There is something called quorum sensing. There is competition between them. Then there is a competition for the nutrients. There's competition for adherence. All these are happening. So how they interact with each other and how they manage to survive there in our gut is important. They form a dynamic and interactive micro-ecosystem which can change in time and scale. So for example, when we are sick, it changes. When we take antibiotics, it changes. When we don't sleep well, it changes. All these problems are there. And it is integrated into the macro ecosystems, including the eukaryotic host, and is crucial for their functioning and health. In the absence of these good microbes, we have a whole lot of health problems. And each individual's microbiome is unique, but with similarities and variations among the subpopulations. So just like the nutrient requirement is different for each one of us and different for us at different points of our life, same way it is to do with the microbiome. The gut microbiome is modified by varying metabolic responses to diet and food components. Disease prevention and progression plays a role. The diet and environment can affect it. Nutrients and bioactive components can affect it. And the synergies and antagonistic interactions, like the chemistry of these components and the concentration of these components, will affect. And many a time we find that these components, some of them have a role only in the gut, but some have systemic effects, which means certain components are absorbed into the system, both produced by the microbe or altered by the microbe. So for example, if you take polyphenols are acted upon by the gut microbes, and some of them will get absorbed into the system. Some others may not get absorbed into the system. And some of these polyphenols will bind to certain microbes and inhibit their growth. So all these kind of complex interactions are happening within our gut. So from here, how do we move ahead? What are the possibilities, or what has actually happened in this area of precision nutrition? So in precision nutrition, the focus is use of genetic and epigenetic biomarkers of individuals or a subgroup of individuals in order to predict the risk of developing chronic disease, in order to personalize their prevention and treatment, and how the nutrients and bioactive components modify epigenetic markers and gene expression. That is the focus of precision nutrition. And the ultimate aim is to use this personal information about the individuals or groups of individuals to deliver nutritional advice so that it is more suitable than the generic advice. So we also take a low fat diet, take a low sugar diet, take less salt, have more complex carbohydrate, eat more dietary fiber. This doesn't work across people, across all people. So we need to modify. So it has to be individualized to establish nutritional guidelines for specific subgroups instead of the conventional population-based advice. So finally, we want to promote and maintain health and prevent disease through the use of the diet. That is the main. Of course, exercise also, but I'm not at all touching or going into the details for that. So the precision nutrition intervention therapeutics is based on nutrients and bioactive compounds, which can modify the epigenetic markers and gene expression, what I mentioned earlier, and how it can help. Prevention and treatment of diseases is possible because it can predict individual risk, it can explain their etiology at least partially, and it can enable us to have a personalized management approach for their nutritional intake. So if we look at the elements, currently this is what we have, more may be added later, we don't know. So you look at, all the orange colored ones, which I put there. Personal data, this we've been doing since so long. Okay, both dietitians, nutritionists, everybody is using this approach. Personal data, dietary intake data, meal content and timing, anthropometric data, physical activity data, clinical and biochemical parameters is the traditional method we've been using so long. So now the additional factors which we are looking at is the metabolomics, microbiome, nutrigenomics, and the genetics. So these factors now we are analyzing in patients, and then we are trying to understand how we can link that to this which we've been using earlier, whether that we can see some patterns and whether these patterns can be meaningfully modified in the dietary approach. So approaches for PM, I just put the major factors again just to repeat and to re-emphasize. So the single nucleotide polymorphisms and other structural variations, DNA methylation, histone modification, microRNA expression. miRNA stands for microRNA expression, gene expression profiles. So these are now what we are paying attention to. What you see on the right side, we already know, but we know much less about the microbiome. All other factors we are aware of. That is the age, gender, ethnicity, pathophysiological status, the nutritional intake, which includes dietary intake, absorption, metabolism, excretion, etc. So here we are looking at the risk prediction. So can we sort of integrate both this set of information? Can we predict the risk which is a person is likely to have? And then we try to provide the nutrition on the genotype phenotype based approach so that we can give a personalized treatment and enhance the responsiveness of the therapy which we are executing. Now here I've just taken some studies and put down together because a whole lot of work is there and details. Again, I've given the references so you can go back, and those of you who are interested can read the details of this. Okay. So some of the associations between variables which have been reported with respect to precision nutrition. So polymorphism in genes related to taste perception, including the sweet taste receptor, TAS1R2, and cluster differentiation 36, which is the CD36, which is found associated with the dyslipidemia. This was observed in Mexican subjects who are consuming high amounts of carbohydrates and fats, respectively. So there is a link to that. Common variants in gene regulating homocysteine metabolism. We all know homocysteine plays a significant role in heart disease, cardiovascular disease. So methylene tetrahydrofolate reductase, MTHFR, and methionine synthetase, which are linked to increased risk of breast cancer in individuals with low intakes of folate, vitamin B6, and vitamin B12. So now we can understand who will be the people at risk. So you can screen for these appropriate markers and then you can give them the advice. And SNPs in the vitamin D receptor, VDR. This vitamin D receptor has demonstrated several SNPs. This has been reported even much earlier, which affect the vitamin D bioavailability associated with osteoporosis predisposition in postmenopausal women who have low calcium intakes. So we know that giving high calcium indiscriminately will not help. Not all people respond to that. So we need to understand why. And we need to use a different approach. SNPs in genes which encode for the lipid proteins like apolipoprotein C3, apolipoprotein A1, which were shown to confer a higher risk on metabolic syndrome in subjects who sort of ate or who complied with the Western dietary pattern, which of course you know is high in simple carbohydrates, high in fat. A genetic variant in the cytochrome P50 family 1, subfamily A, member 2 gene, which is associated with an increased risk of hypertension and CVD in moderate and heavy coffee drinkers. So these are some of the associations. Then examination of the cumulative effect of SNPs on diet interactions and disease susceptibility. These studies showed that macronutrient intake modified the association of an obesity. What is GRS? Genetic risk score. They calculate that on a certain basis with greater values of adiposity. An obesity genetic risk score interacted with the intake of sugar-sweetened beverages and fried food consumption in relation to both BMI and obesity in several cohort studies. This is what they found. And nutrition may directly affect the expression of genes which regulate critical metabolic pathways. So they evaluated the effect of different dietary factors on gene expression profiles. So what are the genes which get expressed for a particular dietary factor and how these are related to disease susceptibilities? What did this show? This showed that subjects who followed a Western dietary pattern which had high intake of refined grain products, desserts, sweets, processed meats, here, the gene expression profile was associated with an inflammatory response. So inflammatory markers went up. Cancer signaling factors went up compared to those who consumed high amounts of vegetables, fruits, and whole grains. So this is the first step. So when we say a disease happens, as all of you know, when we look at the dietary factors, we don't see any change in the individual for several years. Nothing happens. They're maintaining stable weight. Particularly with cancer and some other diseases like even cardiovascular disease or diabetes, we don't know. External symptoms are absent, but changes are happening at the cellular level. And that is what we want to identify and use those to see who are the people who are at risk, and then take the corrective approach or the modification which we desire or which we think will work. And that can also be identified, or we can try and understand that with the help of precision nutrition. Similarly, pathway analysis revealed that high meat consumption was associated with gene networks linked to cancer in the colon tissue. So these are some of the studies. Another very interesting two studies which was reported is the prediction of blood glucose response. All of, just as I mentioned initially, we don't respond the same way to the same food. The blood glucose response is completely different in people, okay, even if they're diabetics, even if they are pre-diabetics, whatever it is. So predicting postprandial glycemic response, 137 individual features were looked at in 800 patients by the ZV et al. In 2015. Similarly, the microbiome-based prediction of insulin resistance by Wu et al., which is a more recent study. So what is the gist of these two studies? Overall, the gut microbiota composition was altered in groups with impaired glucose tolerance, combined glucose intolerance. Combined glucose intolerance is IFG, that is what we call the fasting blood glucose is high, and also the subsequent response to blood glucose or glucose intolerance is high. One is the resistance to the of the liver to insulin resistance in the liver. The other is insulin resistance in the muscle. So both of these, and of course type 2 diabetes, but not in those with impaired fasting glucose. And the abundance of several butyrate producers and functional potential for butyrate production was decreased both in pre-diabetic patients as well as the type 2 diabetes. So this butyrate is very important because it has a lot of functions in regulating the epithelium of the colon. So the way, as you know, the colonites are sort of, they slow off through our feces every 2 days. So the new cells come. And this butyrate is an important regulator of gene expression for the synthesis of those cells or the multiplication of those cells. Multivariate analysis and machine learning microbiome models indicated that insulin resistance was strongly associated with microbial variation. So this is very evident today, that people who have diabetes have a different microbial composition compared to people who don't have diabetes. Therefore, gut microbiota represents an important modifiable factor to consider when developing precision nutrition medicine approaches for delaying or preventing type 2 diabetes. The human microbiome contributes to inter-individual variability, as I mentioned earlier. It also represents a potential target for the management, which could be modulated by therapeutics, dietary changes, use of prebiotics, probiotics, or a combination in symbiotics, and life biotherapeutic compounds corresponding to given species or mixture of species, okay, and microbiome transplants, which is now an FDA-approved method for treating. Now then, those were all how the nutrients affect the gene expression. Here the nutrients and bioactives, how they regulate critical gene expressions. So here we have reports which show that Mediterranean diet reduces the postprandial expression of genes which encode proteins related to inflammation, endoplasmic reticulum stress, atherogenesis, and oxidative stress. Also, high intakes of MUFA through the consumption of olive oil is associated with low expression of genes involved in inflammation and abnormal lipid storage. Epigallocatechin 3 gallate, T-flavin, curcumin, sulforaphane, genistein, these are all, as you know, the various polyphenols, exert anti-cancer properties by upregulating tumor suppression genes and downregulating tumor promoting genes. In addition, they also found that curcumin and resveratrol have anti-atherogenic effect by decreasing the expression of matrix metalloproteinases. So remember, Professor Premavati had mentioned about the metalloproteinases, which are involved in the plaque formation and progression of the plaque. It was noted that apple polyphenols prevented diet-induced obesity through regulation of genes involved in adipogenesis, lipolysis, and fatty acid oxidation. So from this, we understand a whole lot of components which are present in our diet are affecting the way our genes are getting expressed and the way the disease can start or get initiated. It's also been found the precision nutrition can support greater compliance from the side of the patient or the subject. Genetic testing to provide nutrition advice is becoming commercially available and it's gaining interest. Some studies have also shown increased adherence or more effective behavior change in response to personalized approaches. This is an important study in 2017. The Food for Me was a large randomized controlled trial investigating personalized versus generic nutrition advice for including dietary behavior change. After 6 months, they found that the personalized advice groups implemented and sustained more dietary changes thought to be better for their health than the generic group. This suggests that nutrition personalization can improve the health of individuals. One more thing which they found was that when people are given their genetic profile, they sort of a certain sense of ownership. They said, okay, I'm now having this problem. They accept the fact that they have this problem, they have this susceptibility. And because of that, they are willing to take action. Okay, I am different from somebody else. So now I have to take responsibility and I must do something about it. So that feeling also comes in. That's what some of the studies show. Now, when we look at precision nutrition, I thought I should touch upon this because how do you deal with all this data? How do you exactly get all this information? PN approaches can involve the processing of lots of data of different kinds in a way that has not been possible in the past. So when I was talking about the advances in molecular biology and genetics, I thought I should also mention about the advances in this area because this is one area which has helped. Development of big data analytics, cloud computing, artificial intelligence, and machine learning has facilitated such data processing in a way and on a scale which is unmatched by humans. So that is how now we are able to integrate all this information. On one side, you have the genetic analysis or genome sequencing tools, gene expression analysis tools, and then you have the AI. So both of this are being put together along with the traditional factors which we've always analyzed. So complex arrays of factors are integrated to provide precise nutritional advice on an individual or stratified level, which therefore facilitates prediction of postprandial glycemia, triglycerides, whatever it is, whichever disease it is. In these scenarios, the use of sophisticated techniques such as machine learning and deep learning to interpret multiple factors is of great utility. Aside from the final output of PN, ML is also helpful in the collection of data required to obtain the features of this particular model. Common features that are being currently investigated for ML application to acquire data include energy, food and drink intake, physical and sedentary activities across the day, glycemia, and sleep tracking. So they're trying to look at all these multiple factors. Now, normally when we do any study, we are doing all this individually, we're tabulating them in an Excel sheet, and then we're trying to make meaning out of it. Now, in machine learning, this is all made easier. Okay, so advanced analytical tools with respect to machine learning and deep learning have really helped because they are used in studies to identify and classify the variables which are of significance, which are of meaning. So it extracts the data such as gathering the dietary and physical activity data, integrates the features of the model to provide the output, generates algorithms which learns the patterns within that data sets and uses these patterns to make a maximum likelihood prediction about the outcome. So this is another cup of tea, which means there is a great scope for interaction between computer scientists and nutritionists. So just 2 studies have been reported which have looked at the holistic picture, PN models for nutritional management of chronic disease, 2 recommendation systems to provide nutritional support for those with chronic disease have been developed, one by Baker et al., the other by Kim and Chung. Okay. The first one provides nutritional support to individuals with chronic disease in the form of recommendations of suitable dietary alternatives. So they are giving you that. That means you have one component. So instead of that, what components you can take. Then the second one recommends healthy foods based on the user's physical and mental health through dietary nutrition, food preference, and healthcare personal data. What they're proposing is a hybrid model, which combines these factors to ultimately provide a food recommendation on a stratified level with regards to chronic disease status and a personal level in terms of the food preference. So if we do this, they are proposing that it's possible to achieve the objectives of PN. And this performs best by allowing both health information and preference data to be integrated. So with that, so there are so many unknowns, there are so many opportunities and challenges. Where are we? How are we today? So opportunities, the personalized approach is promising because it can reduce the burden of disease compared to the current way we are doing it. Machine learning can be incorporated precision nutrition research with high performance. Precision nutrition researchers have to consider incorporating machine learning into their methods to facilitate the integration of all these complex features so that our final output is more precise. It is more, what shall I say? It is more, it is more authentic in terms of the outcome which it can give us, the positive outcome for the patient. But lots of challenges, because as you know, both these tools, the biotechnology tools which are needed for studying the gene expression patterns, the genome sequencing, metabolomics, proteomics, all is highly expensive. Now the cost has come down. AI is another very expensive thing. So how do we manage all this? The actual process of PN research is riddled with many challenges. It has hurdles to be overcome and must prove itself to be effective and worth the investment before it be deployed. Obviously, only the very developed countries and countries which have good, technical support will be able to achieve this. And although PN approaches show promise in outperforming the population-based approach, this is still not guaranteed. We have a long way to go. And it needs to be tested with rigorous large-scale trials to see really if it will work, particularly in various ethnic populations. What we have now, what the picture shows right now is that, okay, the possibility is very good, but we have to go forward. And whilst techniques for gathering the data and presenting the outcomes can be expected to drop in price and become increasingly convenient, it may take a long time before this is actually feasible. And in a country like ours, where the health sector is partly supported by the government, but most people prefer the private sector. What would be the cost? We have seen in the case of the COVID itself how the vaccines cost differs, how the approach used differs, who can afford, who cannot afford. So this will again, there is a tendency for a sort of inequality for access to health with respect to precision nutrition. Before this approach can be expected to solve the problems of nutrition and health the world currently faces, two important things need to be taken into consideration. The research to investigate the actual efficiency of PN in particular populations in terms of its ability to alter behavior of the consumer or the patient, and the cost benefit analysis. So we're putting in so much. So you can just imagine the cost of research. As you know, to develop a particular drug, even to develop this vaccine, the amount of money which was pumped into it. Finally, how many people really benefit at the time they need. So this cost benefit analysis of the economics is very important for the policy developers and for the researchers. So all stakeholders need to be concerned about this. Then the other aspect, which I think also Dr. Premavati touched upon, the life cycle and metabolic imprinting, because we have the first 1,000 days, all of us know, is very, very critical. So what is the impact of dietary factors in this early phase and late phase? The variability in human development and functioning, when in the life cycle, in which tissues. So this is important because gene expression is so active at that time. Early nutritional events are known to alter an individual's normal development. So this critical window which we have, this metabolic imprinting and programming, which finally decides how our health will be 60 years by the time we are 2 years old, is something which needs to be studied with respect to precision nutrition, or at least the genetic tools. And the lasting impact on the long-term health and well-being of the individual. These are all areas which are still very gray. We have some data, we do not have enough data. So the priorities for research will be to understand the variabilities in metabolic responses to the diet and its components, identifying the variable responses which contribute to health promotion and disease development, big data analytics of large populations and their subgroups with respect to lifestyles, health risks, diseases, drivers of food choices are very important. We all know that certain foods are good, nutrition, balanced diet is good, but how and why are we making particular food choices? Why are we still having this problem? So the biochemical and the behavioral basis involved in food choice is something. Taste is a very key thing. All of us know that. Whatever we tell people, if something is not tasty, they are not going to eat it. They'll eat it once, they'll not eat it later. So tools that can use a systems approach to understand the energy and nutrient balances, these are very significant. Sorry. So PN has the potential to identify people at or groups at risk, and using the dietary composition in terms of the nutrients, bioactives, food choices, and the lifestyle, we can go somewhere. Okay. The way forward is that precision nutrition, if we look at it in a realistic status today, its realistic status today, it is at a nascent stage. It will definitely grow further and wider. It can have a profound impact on individuals and subpopulations' health. We need to apply multidisciplinary approaches if we want to head somewhere in a positive way and in the desired outcome, which all of us want for individuals as well as for society. Thank you very much. I'm happy to answer